Okay. Dear Chairman, dear chairs, actually, dear guests and colleagues, it's a big pleasure to be here, and I'm very honored. And as an outsider, I'm not a researcher working on the obesity part, but I'm in the drug development in general. So, as the first slide, the conflict of interest, I have no potential conflict profile, since I'm the chair for the IRB at Koç University, the member of Ministry of Health Advisory Board for the clinical trials, and a member of UNESCO International Bioethics Committee, actually. So all my research is funded by public funds, actually. So that's what I mean. I start with the question where we are in terms of the personalized medicine. So the overview of what is personalized medicine is going to be the first part, and then is there any experimental model to test the personalized medicine, and to what extent, just a minute, personalized treatment is possible, is going to be the third. And I made this little logo here to emphasize the fact that personalized medicine is a big promise, actually. And I think there's a big paradigm shift is going on throughout the medical community and in the drug research. I will just try to entertain with this idea, okay? Oops. So what is personalized medicine, basically? The new disease taxonomy actually changes the diagnosis, but it doesn't make the treatment personal per se. Okay, so this is something that I try to come up with an idea of having a bottom line sentence at the very first moment. Okay, it is a big pleasure to be here in Vienna, because Vienna, I guess, and I believe, like Eric Kandel in his very seminal book, The Age of Insight. I think the scientific medicine was born here in Vienna by virtue of the fact that the symptoms and pathology actually was linked in Vienna by Rokitansky when he was dean, and Škoda, he was the head of the internal medicine. And at that time, the pathologic, microscopic pathology actually came to the reality, and the scientists and artists, actually, like Klimt, they get together in their homes after hours, and they watch histologic slides. Like this inspired very much Klimt also, in this artwork, actually. So the idea of scientific medicine, actually, is the fact that it is evidence-based. It is evidence-based, and we know that the evidence pyramid and the randomized controlled trials are the mainstay of the scientific medicine to assess efficacy and safety, which is very important. And since the second half of the 20th century, the developments in molecular biology and the deciphering of the human genome and patient profiling has changed the whole scope and the understanding of the randomized controlled trials, anyway. So in the classical paradigm, which was a little bit of, based on a serendipity, find by screening some molecules with one phase, one drug, one indication scheme as a classical paradigm, one drug for all, basically. We have the disease model, we found disease targets, drugs made, and tested on patients, basically. But with the new era, the targeted therapy changed this, and the diagnosis now is pretty much patient-specific. But when I mean specific, it's not just one-to-one, personal, of course. Nevertheless, in this situation, the patient profiling actually defines the disease, and the disease taxonomy is a little bit changed, and I think it's going to be more and more in the years to come. And we try to have some specific drugs for specific conditions for each drug. The main distinction of this new system is that new evidence gathers in all steps of this clinical trials, so that what we have done is to have many interim analysis and feedbacks over this protocol, so that the protocol is changing constantly. So it is going to be a very dynamic kind of situation. So we obtain success when outcome measures are clear. Like, after the profiling, we reach the treatment. In cancer, it is very straightforward. We identify cancer cells and kill. So that's a very, straightforward kind of situation. Biomarkers play a very important role, and also measurable parameters that are very important. An example, a seminal example is with the BCR-ABL mutation, which results in the leukemia, and when we attack proteins formed by this mutation with imatinib, we obtain a very big breakthrough kind of result in the clinical medicine. But in complex diseases, it's totally a different story. So I took this photo from a park in Istanbul, actually, and this is the metabolic syndrome monument, by the way, and it seems that the surgeon god actually kills the metabolic syndrome, like all surgery does actually. Here the problem is the fact that in complex diseases you don't know by drug terms how to attack. So the artist apparently, inspired from the De Fronzo's paper, in 2009 paper, showing that the main ingredients of hyperglycemia named as the ominous octet, and this ominous octet being a monster, actually. Okay. In complex diseases, the modeling is a problem, like metabolic syndrome, obesity, type 2 diabetes, but also in hypertension and in many other complex diseases. And they are all multifactorial, complex, and difficult to attack. So what we do actually is, we have now a big tool named as genome-wide association studies. We detect SNPs and copy number variations, indels, and so forth and so on, all kind of small mutations, and try to associate with this to different phenotypes, like fast and slow responders and acute reactions, and try to cluster off-target responses with adverse effects and lack of efficacy. So this is a straightforward kind of paradigm that we use all the way for the drug development. But let's give this example of an obesity example. The FTO gene variant, we know that there are many copy number variations, and these people are obesity prone, and many studies show very clearly that they show a preference for high energy foods and hyperphagia. Unfortunately, in a new paper, from almost 8 years after Frayling paper in Science, Livingstone et al. In obesity reviews show that it's very difficult to prove in large studies the assumption that they made in their initial paper by saying that they have preference for high energy foods. So this is a very important thing that we should keep in mind for all complex type studies, and I will just develop and evolve over this idea. It's not easy to put the causality here. Okay, why? Because there are many flaws in the clinical trial design, like reporting the dietary intake for each and individual patient. And the other thing is, lifestyle changes also the course of the disease. So this is also something that we are struggling to deal with. Another example with Prader-Willi syndrome, it's a complex genetic disease. We know the genetics very clearly. It's a uniparental disomy of the chromosome 15. And we have also a very accurate, almost 99% accurate diagnostic test. And we also have clinical experience with many phenotypes, with very rich clinical features, and many clinical outcomes. So those clinical outcomes also not easy to discuss. The sole approved treatment is the growth hormone therapy, and unfortunately no successful treatment for hyperphagia. Hunger is a difficult signal for analysis here, and there's no biomarker to measure for the hunger, actually, and different phenotypes were pooled. Then social support and quality of medical care also changes the outcome, and it's a very rare disease, so the number to reach to attain significance is not easy. So when I check the trials that involve eating in clinicaltrials.gov, I just focused on phase 3 because this is the critical time when all industry and academics think about the commercializing the drug product. Out of the 298 studies that involve the eating behavior, hyperphagia type of eating behavior abnormalities, only 33 reaches the phase 3, and now currently there are 10 are active. And if you look at the drugs, dasotrelin, diazoxide, intranasal oxytocin, and liraglutide, only 2, diazoxide and oxytocin, are being tried on the Prader-Willi syndrome. And if you look at the terminated ones, there are many of them that are not even reached to the end of the trial for obvious reasons that I don't have time to discuss now here, but we may discuss later on. And none of them for Prader-Willi syndrome. So it's very difficult to do clinical research in that terms, actually. So the path to personalized medicine, I think, to sum up, we need the new smart trial designs like basket or umbrella trials or N of 1 trials. This needs to be very dynamic, and frequent interim analysis are key. Genetic variability must be considered, different ethnic groups. Small numbers are no good for the genome-wide association analysis as well. The consent becomes a very important issue. There's a big discussion within the medical community as well as in bioethicists, blanket versus specific consent, and with this new personal data rules and laws, it's getting to be very difficult. The other thing is the genetic data is not included in personal health records. It's considered as a personal, information, which is not health information, because even when planes crash, we identify the identities with the DNA, so DNA cannot be anonymized in that sense. Okay, and we need to engage also patients in clinical trial community. I just will mention a little bit later. So the lessons learned, retrospective analysis by taking genome data into account is a crucial thing, but this is possible if the samples were collected beforehand. So you need to have a consent to do this. And the consent may not be so easy to get because we don't know what our research will lead us in the future. The analysis of adverse effects may benefit from such opportunities, and drug repurposing in that matter is a very important thing. And difficult to meet the expectations of the patient's families. The consent taking, as I said, is a big problem, and the cooperation with mentally challenged patients is difficult. The other, the thing that I want to point out here is that the low genomic literacy is a risk factor, as Nakamura et al., I think, brilliantly showed in the hypertension and obesity studies. And it's not easy to improve the level of knowledge with this complexity of the genome literacy, basically, especially in the patient groups. And funds are limited because drug companies don't like small groups to get the results. And the support for basic research is very critical, is obviously, okay? So obesity is a very hot research area. And if you look at the experimental models, you see when we get to know the DNA, if the gene is unknown, we put it in a mapping unit and we start cell biology experiments together with animal experiments and biology of disease and biomarker identification. I think this one is going to be, the whole circle is very important to reach the new drug targets and new therapy modalities. And in the end, this translational medicine clinical trials relationship also is very critical. And if you look at the experimental models, as we go to the in vitro studies, the throughput is very big, actually, owing to the molecular biology and cell culture experiments, and the cost is quite acceptable. But as we go up to the ladder to in vivo studies, and starting from zebrafish to non-human primates, the relevance is getting increased. Nevertheless, the cost is very big a problem too. And in biotech terms, a major breakthrough has been happened in every 10 years. We started with the 1980s with transgenic techniques. Leptin knockout mice was very big issue at that time. But today, in 2010, we have the gene editing with CRISPR-Cas system. Gives us a very wide application areas. And just recently, in 2018, there's a leptin and leptin receptor gene editing paper from a group from Korea, which also allows us to tell you about reverse genetics rather than the forward genetics. I think this is going to be a big breakthrough in the years to come in complex diseases. So how feasible is it? So if you look at today's situation, the individual is in the beginning, at the center here, and there's a huge accumulation of big data, which makes the individual as transparent as can be over the years. Nevertheless, the whole system that contains the data actually is getting more and more opaque and not so transparent actually. So there's a big concern in many ways to manage this big data, which is very important for the hypothesis forming. And those hypothesis testing may yield to several different molecules that may be termed as personalized medicine. But the personalized medicine here is not the personalized medicine as we think of a tailor-made suit, actually. This is pretty much clusters of groups. And these cluster groups, some of them could be much more advantageous than some others, actually, in terms of the medical treatment and the outcome. This is also something that we should think about in the future, because the value here depends on the patient outcome versus cost, and it's obviously that there's a limitation factor in that the cost should not exceed to a certain limit in order to be applicable to any person in a system, actually, within the countries and in the global scale as well. Okay? So if you see the big picture now with the risks and benefits, I told you that the 4 Ps of the personalized medicine is the personal, preventive, predictive, and participatory. And if you look at the preventive, predictive, participatory part, this gives you too much responsibility for an individual. What I mean for that, it could become too obstructive. For instance, if you get the whole authority about your genome, actually, then people may say that, okay, you have the risk to become obese, then if you become obese, it's your fault kind of situation, okay? And this may have, signal an emergence of an authoritarian medicine, like your gene is your fate and your fate is sealed, and it's pretty much like that. And data repositories actually contain very highly sensitive data, and the different stakeholders may have a very different point of view about these repositories, like insurance companies, drug companies, and governments and individuals as well. So another important issue is the left behind populations, because we don't have the ability, we will not have, at least in the midterm, a possibility to address to all kind of people. So it's going to be subsets of people that are more advantageous than the others, and the left behind populations could be a big problem. And I also think, as a cautious physician, applications of a new technology may have unexpected outcomes, and social impact needs to be discussed. On the other hand, there's a very important positive side, which enables also the patient to seize its fate, because it knows through the DNA-based technologies what he needs to do, actually, and some other people may also benefit from that fact either. So the question is, how fast shall we adopt personalized medicine? From an individual context, it's the beneficence, justice, and autonomy is the main tenets of the bioethics, basically. And from the social context, the political decisions, equitability, and affordability are important, ideas. Unfortunately, or far fortunately, because humans are social animals, social context sets the ground rules. So this may have a very disparate kind of applications and management in different parts of the world initially. But I think the world needs to be discuss these all together. I finish my talk by just putting this question, which precedes which. We have the science which really works for the beneficence of the humanity, and also, the most importantly, for the veracity, because the real world, the reality actually is determined by the science. So even if we don't like the end, if you become pretty much sick or whatever, bioethics, on the other hand, deal with the equity, justice, and autonomy. And sometimes, in order to have autonomy, you need to have the full veracity, actually. So if you don't know anything, you cannot have an autonomous kind of decision, actually. Too much emphasis, on the other hand, on genetics may put social determinants of health in peril and redefines patient-physician relationship. And I think we need more and more public deliberations, and the debate on personalized medicine should not be limited to expert panels, actually. And for these, I really thank for your attention, and with my campus in overlooking the access.
Thank you very much for this very interesting talk. Again, we have no discussion.